Hello class 12 science students. I hope all of you are studying and you are examining or you are watching the video properly. Okay. If there is any doubt, please get a query through WhatsApp. Okay. Or if there is anything, you can write up in the comment box. Today we are going to discuss very important topic, that is translation. Okay. Now, in the process of translation, you know that mRNA, your mature RNA, will be synthesized to protein. Now today we are going to discuss how proteins are synthesized in our body, because proteins are required for the functions. Okay, it may be for the immune system, it may be for the enzymes production. Okay, we need proteins so that there will be a normal growth and development. Okay, or in another way, the development of immune system to fight different kinds of diseases. So the protein production is very important. Before undergoing the step of this process, let's see some of the basic requirement. Okay, the first one is initiation codon. We have already discussed initiation codon. Okay, very important codon, that is AUG. AUG, AUG. So this is a codon which code for methionine. Methionine. The first amino acid that comes, just say that this is your protein. And as I said, that proteins are made up of amino acids. This is your protein. Okay. So the first amino acids always is methionine, because that's why we say that OG is a codon that code for methionine, and it also acts as a initiator point or the initiation codon. That's why we call as initiation codon. Okay, because from this point only the process of translation starts. So it has a dual functions. So no other codon in the genetic code will have dual functions except O. Okay, so just say methionine, another valine, okay, isoleucine, leucine. So when many amino acids combine, you get a protein. But to start, we need initiation codon, and that is O. The second one is termination codon. We have already discussed that termination codon, or we call stop codon. So these are the three important stop codon. In the previous class, I've already shown you how to make genetic code, and I said that these are the three UAA, UAG, UGA. Okay, it has name like amber, ochre, and opal. So these three are said to be stop codon or the nonsense codon. The third thing that you have to know is about ribosome. As you know, ribosome has two subunits, okay, large and small subunits. Now if you see prokaryotes, I'm just writing as P. For prokaryotes, it is 70S. Okay, it means that it is divided into large subunit as 50S and 30S. We are not adding or subtracting. Okay, here just now in this, the prokaryotes have 70S. Okay, so it is 50S, that is large subunits, and for 30S it is small subunits. Whereas for eukaryotes, okay, it is 80S. Okay, it means that it is 60S. Okay, as a sedimentation coefficient, that will be considered as large subunits, and 40S. Okay, this is for small subunits. So we are talking about the subunits of the ribosomes. Okay, the fourth important point is where this process takes place. So this process, the synthesis of mRNA, from mRNA to protein, the synthesis of your protein, it takes place in cytoplasm. And Remember that for DNA to RNA it takes place in nucleus, right? Nucleus, okay? This is for mRNA to protein is in cytoplasm. This whole process will take place in cytoplasm. Next, number 4 is magnesium ions. For the process of translation, we need Mg plus. Textbooks, if I write Mg plus plus, the same, okay? Magnesium ions are required. And the 5th important one, we have only one enzyme, okay, responsible in the process of translation. That is amino acyl tRNA synthetase. Amino acyl tRNA synthetase. So when I'm explaining when I will explain the process of translation, I will just say E. I will just represent as E as an enzyme. Okay. Now this enzyme, okay, it has 2 subunits. I'll just explain here. Just say that this is your amino acyl tRNA synthetase. It has 2 subunits, means 2 area, okay, 2 division it have, and one side just I'm writing as AA. So in this side amino acid will go and bind, okay, and the another side will be for tRNA. So these are the 2 functions of your amino acids. One is it is helping for activation of amino acids, okay, it activate the amino acids, and the second is it also bind the tRNA and it transfer the tRNA, it transfer the amino acids to tRNA, okay. Let me explain again. It has 2 subunits. One is this area, one subunit is for amino acids, for the activation of amino acids. And the second is for the tRNA, where tRNA will go and bind, and then the amino acid is transferred to the tRNA, okay, with the help of amino acyl tRNA synthetase. Now we're going to discuss how those amino acids are activated. See, there are 20 natural amino acids. 20 natural amino acids are present in the cytoplasm. They are freely moving, okay, and they have to be activated. Now, how to go for activation? The first one is we require ATP. We need adenosine triphosphate, and we need enzymes, okay, E for enzymes, and then we need amino acids. See, amino acids are moving in the cytoplasm freely. So this is called as activation of amino acids. Which will be the first amino acids to come for activation? I hope if you guess, you know it. This is methionine. First amino acids will be methionine. Don't forget this, okay? Now, this enzyme, after activation, just say that ATP, enzyme, and how they get activated, okay? So, this is your amino acids, okay? Then you get your AMP. AMP means adenosine monophosphate, okay? Then enzymes, okay? Plus pyrophosphate, means 2 phosphate group has been released and a bond has been formed, okay? Now, once this is called your activation of amino acids. Once amino acid is activated, now it has to transfer to tRNA. Now your tRNA will come. So I'll just make your tRNA like this, okay? Your tRNA will come and bind to this enzyme, right? So this tRNA has a empty codon loop. So now this is your enzyme. Your amino acids is there, right? AMP is there. We need energy for bond, okay? And then for the bond formation, okay? Now this amino acids, and then here it will be your tRNA. Now these amino acids will be transferred here, okay? As I said before, also in the 3 prime. Okay, for example, this is your so this is your 5 prime, 3 prime, right? So in this position where there is a hydroxyl group, okay, in that position your amino acids will bind. Okay, so in that way, once the activated amino acid is transferred to the tRNA, now what will happen? What will be the result? Okay, you will get tRNA, amino acids with tRNA plus AMP will be released, and even the enzymes will be released. Okay, so now this activated tRNA, or we say the amino acid with tRNA, okay, we call that as formulated, okay, formulated formulated tRNA. It is no more as tRNA, but it is said to be formulated tRNA or methionine formulated tRNA. Why methionine? Because the first amino acid is methionine. Now, this is about the process of the activations, how the amino acids are activated. Now, the process of initiation and elongation. Number 2, okay, where initiation and elongation will be discussed in point number 2 itself. See, the ribosome, as I said, it has 2 subunits, but the larger subunits, so I'm just making, so it has 3 sites. E is called exit site, P site, P is called baptidial site, baptidial transfer, or donor site we call sometimes, and A site is called the acceptor site. Now just think in the cytoplasm where tRNA is bringing amino acid continuously, okay, depending on the requirement. Okay. Now on the other side I will make RNA. This is your RNA, 5 prime to 3 prime. Now the process is going to begin. Okay. So where the process will start? Obviously where there is a O, the initiation codon. Okay. This is a nitrogenous base. For example, there will be A, D, C, D, A, G, G, U, U. Okay. Many nitrogenous bases will be present in the mRNA molecule. So wherever there is a O, what will happen? The smaller subunit of ribosome. See, ribosome have, this is a large subunits, and there will be another smaller subunits. Smaller subunits. So these smaller subunits will go and bind. Okay. Once it binds, what will happen? Your tRNA will come now. So this tRNA is carrying your amino acids. So the tRNA, if there is A, then it will be U. It has an anti-codon loop, right? So if U, A, C. Okay. Now once the tRNA comes and bind, what will happen? The ribosome, the larger subunits will come and bind. Okay. So now the process continues. So it brings amino acids. Now as the ribosome started moving, it means that this ribosome will move to this codon. Once it moves, what will happen? It will bring another amino acids, right? It can be any other amino acids depending on the codon. Now if these ribosomes, if it move here, you see my ribosomes are the protein manufacturing unit. So they have to move. As it moves, what will happen? tRNA will come. Now you may be thinking which tRNA will come? The activated tRNA, okay? Depending on the requirement, the different types of tRNA based on the codon. If there is GUU, so there will be another tRNA which have CAA, which have an anti-codon loop. So they will come and bind here. So as the ribosomes start moving, different tRNA will come. They will bring the amino acids. Okay, see, they are bringing the amino acids. And just think that these ribosomes have reached here. Okay, somewhere here in the AAC here. Okay, these are large subunits and there is a small subunits. Just say it reach in the termination codon where the process will stop. So I'm just writing, these are the amino acids. Okay, just a long amino acids is formed. So these are all amino acids. I just made a amino acids. So these are the amino acids. Once it reaches to the termination codon, it will stop. The ribosomes will move away. Okay, smaller subunits will move away, and then the process will stop. Now, one thing that you have to remember is that what we discuss about E, P, and A site. Okay, in E site, the first tRNA. Okay, the first tRNA will always come at the P site. Okay, it will always come at the P site. The next amino acid will come to the acceptor site, because as the ribosomes start moving, what will happen? The next amino acid have to come, and that will bring by tRNA, right? So the first amino acid, the first tRNA will see, ribosome is a big subunit. It will capture. Okay, so P site will be the site where the first tRNA will come. After that, the next tRNA will come at the A site. And where this tRNA will leave? Exit site. Okay, once they bring the amino acids, for example, this was brought by this. So after the next will come, right? So this was given by this another amino acids by tRNA. This tree another, okay, another tree. So in the P site, the first will come, but after that, the next tRNA, okay, will come at A site. Okay, so once that bond, so this is called as your peptide bond. Okay, the bond between two amino acids is peptide bond. Okay, so after that, as the process keep on going on, the amino acids started joining with one another, and they form a protein. Okay, and certain modification will take place later on, depending on the requirements of our body. Okay, so this is a process of translation. The first one is activation of your amino acid with the help of ATP and your enzyme amino acid tRNA synthetase. Then activated amino acid is transferred to your tRNA, and we call as formulated tRNA. Okay, now the third is the larger subunit. The smaller subunit will go and bind to the mRNA. Then tRNA will come. Then larger subunits of ribosomes will come and attach. And as the ribosomes started moving, the amino acid will start forming bond. And in that way, the proteins are synthesized in our living system. So for today's class, I will wind up, and I believe that this topic you have understood. Okay, and if there is any doubt to be cleared, please ask with your query and clear your doubts. In the next class, we're going to discuss about the DNA fingerprinting, some of the applications, okay, like human genome projects, okay, and lag operand. Thank you.